Well, welcome everybody to Grand Rounds. I wanted to introduce our speaker, Dr. Michael Hsieh. Michael has a very interesting background. He was an undergraduate at Stanford and then went to Thomas Jefferson, both to get his MD and PhD, with his PhD being in microbiology and immunology. And then did his urology fellowship at the University of California, San Francisco, then went to Texas Children's Hospital for his pediatric urology fellowship, and then back to Stanford, where he was on faculty for about 5 years as part of their urology group. And then in 2014 was named the Stilwell Endowed Director at the Biomedical Research Institute, which is in Rockville, Maryland. And Dr. Hsieh has a number of research interests that range from robotics to immunology to the area that he's going to speak to us about today, which is the schistosomiasis. And looking at the connection between them of immunology, cancer, and the schistosomiasis. So without further ado, I'd like to turn it over to Dr. Hsieh.
Okay, can everyone hear me? This microphone working? Great, okay. All right, well, good morning. So let's start with the case. This was an actual patient that one of my practice partners saw just a few weeks ago. 9-year-old boy with gross hematuria with no pain. Oh, and by the way, he lived for a year in Mali. So the patient underwent a very extensive negative workup for hematuria for a child. And ultimately, my practice partner took this patient to the operating room to perform cystoscopy, really not expecting to find anything. But she saw this, okay? So these are sandy patches. You see these little nodules, and overlying these sandy patches are erythematous areas of the urothelium. So she biopsied these areas, and this is what she saw on low power and high power views, okay? And what are these objects? Anyone? Eggs? What kind of eggs? Schistosomiasis. How did you know that? Okay, so schistosomiasis is certainly something that we see here in DC, especially given how international we are. And certainly it has global relevance. So I'm going to show a series of short videos to highlight the life cycle, and then we'll talk about its relevance to pediatrics. So schistosome worms, male and female, will lay in copulo, as it's called. So the female worm is much thinner and lies in this gynecophoric canal of the male. So they're clasped together, very romantic. And they'll lay eggs, thousands each day, in the portal system if it's a hepatoenteric form of schistosomiasis, or in the case of schistosome haematobium, which causes urogenital schistosomiasis. These worms will migrate to the pelvic veins, and there they'll lay eggs in the bladder and other pelvic organs. So about half of the eggs that are laid in the intestines, in the case of hepatoenteric schistosomiasis, or the bladder in urogenital schistosomiasis, will be excreted in the urine or stool. And once they hit fresh water, these eggs hatch, releasing the Miracidia within. And the Miracidia are heavily ciliated and very motile little organisms. And this enables them to infect snails. So this is a head and tentacle of the actual snail being infected by Miracidium here. And you'll see the high power view in a moment, okay? So once inside the snail, the Miracidium developed into Sporosis and then into Cercariae. So Cercariae are the infectious stage for humans. They look like little torpedoes, almost sperm-like with a forked tail. And the heads of these Cercaria are barbed First of all, the Cercariae are released en masse from snails. The heads are barbed, which allow them to stay in human skin when they burrow through, and there's these acetabular glands that contain enzymes specifically evolved to dissolve human skin. So this Cercaria is preferentially burrowing into the base of a hair follicle and digesting its way through. Okay, and this is actual video footage of mouse skin being penetrated by a Cercaria. Okay, so just to recap quickly, as I pointed out, adult worms, in the case of S. haematobium, will lay eggs in the bladder and other pelvic organs. In the case of S. mansoni or S. japonicum, the two main species causing human hepatoenteric schistosomiasis will lay eggs in the liver and intestines. The eggs are released in the fecal and urinary streams. The eggs hatch to release Miracidia. The Miracidia infect snails. The Miracidia then develop into Fibrosis, followed by Cercariae, and the Cercariae will infect humans, completing the life cycle. So schistosomiasis is truly what I consider a childhood infection with lifelong consequences. So if you look at peak prevalence, it's really during early childhood, peaking at between 10 to 14 years of age, but there are chronic sequelae that often evolve over decades. And pathology that evolves is almost entirely due to the fact that eggs are highly inflammatory and will induce granuloma formation. And I'll emphasize this throughout the talk. So for instance, if enough egg granulomas develop in the liver of a heavily infected individual, that can lead to periportal fibrosis, also known as pipestem or Simmer's fibrosis. You see this? This looks like a pipe that's been cut in cross-section. And this is a spleen of a young Brazilian who had very heavy S. mansoni infection. So you can also get end-stage liver disease from this fibrosis and sometimes even cirrhosis with development of varices and splenomegaly. This is all due to egg-induced inflammation. And if the eggs are ectopically deposited in the pulmonary vasculature, you can even get right-sided heart failure due to the inflammation associated with this. Now, in the case of urogenital schistosomiasis, there's a really wide range of complications that can be associated with infection. So for instance, there's been some estimates that there's almost 10 million people who have reflux as well as obstruction due to fibrosis in the urinary tract. This is an intravenous pyelogram of a patient who has heavy infection affecting the left side of the urinary tract, and that's why you see severe hydroureteral nephrosis. There's almost certainly bulky granulomas obstructing the ureteral vesicle junction on this side. So the same paper estimates that as a result of end-stage renal disease from obstructive fibrosis, there's perhaps 150,000 deaths that occur from urogenital schistosomiasis each year. Now, interestingly, it's also thought that S. haematobium infection somehow increases HIV risk in the case of female genital involvement. And this is a colposcopic view of a cervix of a girl with heavy S. haematobium infection. And here's the cervical os, and you can see all these very erythematous areas. This represents hypervascular sandy patches that are very prone to contact bleeding, which is probably at least one of the mechanisms by which girls and women with FGS have a tripled or even quadrupled risk of contracting HIV. And finally, S. haematobium infection is known as class 1 carcinogen according to the WHO, which means that they consider it definitely carcinogenic. This is a cross-section of a bladder that has squamous cell carcinoma associated with urogenital schistosomiasis, and these are keratin pearls, which are pathognomonic for squamous cell carcinoma, which at least in the West is a very unusual histologic form of bladder cancer, but in urogenital schistosomiasis endemic countries, this is a quite common form of bladder cancer. So to really drive home the concept that schistosomiasis has a really significant impact, again, due to egg-induced inflammation. If you look at the Global Burden of Diseases study at the 2010 data, globally, the greatest causes of years lived with disability are things like lower back pain, depression, iron deficiency, anemia, and so on. But if you look at sub-Saharan Africa, in contrast, the top 10 causes of years lived with disability include schistosomiasis, which is rivaling malaria and HIV. So schistosomiasis disproportionately affects children in particular, and this is due to a number of factors, including environmental factors, for instance, lack of access to clean water and climate locally, which drives people to any water source that they have available in tropical climates. Biological factors such as age, as I showed you earlier, gender, and any past infections. So there is acquired immunity over time, but it can take a long time to develop, and some patients develop life-threatening sequelae before they acquire immunity. And then finally, there's a lot of social factors that drive schistosomiasis and its effects, including malnutrition, which of course is immunosuppressive, lack of literacy regarding the schistosomiasis life cycle, and the cycle of poverty, which in itself is perpetuated by schistosomiasis. So what do I mean by schistosomiasis perpetuating the cycle of poverty? If you look at children in Tanzania, shown here, up to 40% in some areas are stunted. Now, granted, this is multifactorial, but many studies, and a few of them are listed here, have shown that a significant contribution of child wasting and stunting is due to schistosomiasis. And again, this is driven by egg-induced inflammation and anemia. If you even do very simple tests in the field, like shuttle runs, as shown here, you can easily quantify the negative impact of schistosomiasis on physical fitness in children. Again, this is driven by egg-induced inflammation. And moreover, in school, behavior, academic achievement, and attendance are all negatively impacted by schistosomiasis, as reported by several papers shown here. Again, this is egg-driven. Now, unfortunately, there's only one WHO-approved drug, which is praziquantel. Okay, anyone know what this is? This stick here. Anyone been in the field? So this is a dosing pole for praziquantel, right? You know, you can't have like fancy scales in the field, so they just measure how tall a child is, and based on that, they give the praziquantel dose. Okay. Now, praziquantel is problematic for children in particular because it's a horse pill. This is pretty close to the actual size of the pill, and it's bitter tasting. Okay, so of course, children are resistant to that. Fortunately, there's a pediatric praziquantel consortium that's actively developing a more palatable formulation of praziquantel, but we're not quite there yet. And on top of this, there's no vaccine, and really control of schistosomiasis in many countries is being driven by mass drug administration, where you do random sampling of a small number of people in a given village, and then based on the prevalence of that small sample, you treat the entire village or the entire associated school, okay? And it has its pros and cons a little bit beyond the scope of this talk, but ultimately, MDA relies upon diagnosis of schistosomiasis. And in fact, in some countries, such as Brazil, you can't give MDA. You have to diagnose on an individual level whether someone has schistosomiasis before you can even treat them, okay? So diagnosis of schistosomiasis essentially consists of counting eggs in stool, also known as the Cato-Katz test, or in urine, that's a syringe filtration test, okay? And, you know, these young gentlemen look happy right now, but that's probably because they just started their day. Imagine 1,000 samples later, okay? So really, the current gold standard for diagnosis of schistosomiasis is counting eggs under a microscope, which is mind-boggling. You know, it's incredibly time-intensive. It requires trained personnel. It's actually expensive when you factor in the fact that you need microscopes with a reliable power source. That's a big issue in the field. Slides, and even things like petrol to transport all the equipment and whatnot. And finally, a single egg count is actually highly inaccurate. You can miss up to at least a third of infections with one count. And so the really good field studies have to rely on 3 separate samples for each patient. So diagnosis is a huge issue. Now, there are some emerging diagnostics that are focusing on detecting worms, and they are promising for that. But there's a particular issue with that, namely that you can have 2 children, for instance, with the exact same worm burden, but if child A was infected 10 years ago and has not been reinfected, and child B was infected just a year ago, they're going to have very different egg outputs because the worms are known to senesce, even though they can live for years to decades, but gradually over time their egg output decreases. So I would argue there's actually a need for what's known as morbidity assessment tools so you can measure egg burdens and the impact of schistosomiasis on an individual patient. And this is especially important for countries that are working towards elimination of schistosomiasis and will eventually require diagnosis of morbidity on an individual patient level. So to address this need, my lab's focused on a particular molecule known as the Interleukin-4-inducing Principle from Schistosome eggs, also abbreviated as IPSE. And we're interested in this molecule in particular for a number of reasons, but at least from the point of view of diagnostics, because it's one of the most abundant egg-secreted proteins. So if you want an egg-oriented diagnostic, it's a good bet. So we develop primers that are specific for the S. mansoni ortholog of IPSE, as well as a set of primers specific for the S. haematobium ortholog of IPSE, and then a third set of primers that are bispecific that can detect either species form of IPSE. So what we did first, this is the bispecific set of PCR primers, and I'm showing you here that the cycle threshold by PCR correlates very well with the input egg RNA. The other primers worked very similarly. We then titrated known amounts of eggs into mouse liver tissue, in other words, sort of a spiking experiment. And when we compared egg counts to the input liver RNA, again, we had good correlations. And this was true not only for the bispecific primers, but also the other primers. And this also worked well when we took 10 healthy individuals, volunteers, urine specimens, perhaps lab personnel, and spiked in S. haematobium eggs. The primers worked very well in this setting as well. So next we asked if the duration of infection, because over time an infected organism will accumulate more eggs in their tissues. We asked if duration of infection will correlate well with our PCR assay. So we infected mice with the same number of Cercariae, but then sampled them at different time points post-infection, performed egg counts and PCR, and looked at their correlation. And as I show you here, the correlations were fairly good, and the other primers worked similarly well. We also asked if the intensity of infection, as measured by number of infecting Cercariae, would also correlate well in our PCR assay. So we titrated the number of Cercariae used to infect mice and then perform egg counts in the PCR assay. And correlation, again, was quite good, and this was true with the other primers, and we also did this with stool, which also correlated well. So we think this PCR assay is promising as a quantitative egg-specific diagnostic that may be a good morbidity assessment tool, which is a real need in schistosomiasis control. We do need to test infected human stool and urine, which is ongoing with some of our field partners. And we'd like to compare our diagnostic with some of the emerging diagnostics in a head-to-head fashion. And finally, a field version where we could do isothermal amplification, for instance, right? Who wants to carry around a thermocycler in the field? That would be a very desirable feature of this assay. So that's some of the diagnostics work we have going on in the lab. I'd like to shift gears a little bit and talk about IPSE in a different sense. So these unfortunate children are holding up their own bloody urine samples, and of course they have S. haematobium infection. Now, even though hematuria is one of the hallmark signs of infection, the truth is on a day-to-day basis, most people with S. haematobium infection don't have gross hematuria, okay? So that's kind of strange if you think that each worm pair can lay thousands of eggs in the bladder each day, and half those eggs are making their way through the bladder wall. So why isn't it that everyone has horrible hemorrhagic cystitis? Well, we hypothesize that maybe the parasites making factors that help balance passage of eggs through the bladder wall, which is necessary for the parasite's life cycle to continue, against outright killing the host, which is ultimately not in the parasite's interest, right? And so we hypothesize that in particular that maybe IPSE is one of these factors. And I'll get back in a moment to why we thought IPSE could be this key factor. First of all, a former MD PhD student of mine, Luke Pennington, cloned out the S. haematobium orthologs of IPSE. This is just a dendrogram showing the genetic similarity, sequence similarity between what we call H-IPSE or S. haematobium IPSE and M-IPSE, which is S. mansi-IPSE. As you can see in the upper left-hand corner, the amino acid similarity is not real high between these two sets of orthologs. And this suggested to us that perhaps since S. haematobium causes urogenital schistosomiasis and S. mansi causes hepatoenteric schistosomiasis, that there might be some tissue-specific effects that have evolved from these orthologs. And I'll show you why we think that's important in a moment. So we've done a lot of characterization of these H-IPSE orthologs in comparison to M-IPSE. I won't drill down really deep into the basic science data, but anything I show you here in red font is shared between H-IPSE and M-IPSE. So for example, both sets of orthologs combine immunoglobulin, and through that action will induce basophils and mast cells to secrete IL-4, and that's where IPSE gets its name, Interleukin-4-inducing Principle, okay? Now, M-IPSE can also activate B regulatory cells. We don't know if H-IPSE does that, but we do know that both H-IPSE and M-IPSE can bind and neutralize activity of a whole bunch of different chemokines. So you can't tell, an emerging theme here is that IPSE is very anti-inflammatory. And finally, perhaps most amazing of all, the IPSE proteins have a nuclear localization sequence that allows them to get into host cell nuclei and change transcription of host cells, okay, which is a really weird and unusual property for a parasite protein. So this made us start thinking, instead of a standard microbiology lab, thinking about developing drugs for bugs and shifting to drugs from bugs, okay, as these emojis show you here. So what do I mean by that? The first disease that we looked at was hemorrhagic cystitis. Has anyone had a patient with hemorrhagic cystitis? Okay. So this is a devastating complication that can be caused by prior radiation as well as viral reemergence in immunosuppressed patients, and certainly from alkylating chemotherapy like ifosfamide, cyclophosphamide. This is the structure of ifosfamide, and then this shows a bladder that had to ultimately be removed because of refractory hemorrhagic cystitis. Now, I think many in the audience know that mesna is used to try and prevent these complications, but it doesn't always work, and it can have its own side effects. So I was interested in this disease because as a pediatric urologist, I had patients such as this one. So this was a 3-year-old's abdomen, And this 3-year-old looks like he's pregnant because his abdomen is distended from a bladder being completely full of clot. So this is just some of the clot that I evacuated from this poor child's bladder during one of his many trips to the operating room. And of course, he had hemorrhagic cystitis caused by prior chemotherapy. So this is a really horrible disease, and we don't have good management options for it. So the specific reason that I was interested scientifically in this disease is that others have described that giving recombinant IL-4 in this setting, at least in mice, helps alleviate disease. Unfortunately, giving recombinant IL-4, just like giving almost any other high dose of recombinant cytokine, is highly toxic. But if you remember, the IPSE is interleukin-4-inducing. So hopefully you know where I'm going. Namely, we wanted to address the question if IPSE could prevent hemorrhagic cystitis. So the way we did this was to administer ifosfamide to mice. Some of the controls we ran were mesna alone or recombinant IL-4 alone. And then, of course, H-IPSE, for instance, so wild-type H-IPSE with ifosfamide, a nuclear localization sequence mutant of H-IPSE that can't get into the nuclei of host cells with ifosfamide. As I mentioned, IL-4 with ifosfamide. 3 doses of mesna, which is standard clinically with ifosfamide. The wild-type H-IPSE with a neutralizing anti-IL-4 antibody. So the point of this was to see if there was any IL-4-dependent mechanism for H-IPSE. And then the NLS mutant with anti-IL-4 antibody, as well as ifosfamide-only controls and a saline control. So I won't show you all the data, but some of the more striking findings. So this is a section of a mouse bladder that's been treated with saline. You see a normal urothelium and a lack of edema and no hemorrhage. In contrast, after a single dose of ifosfamide, you see all this loss of urothelial integrity. There's hemorrhage and epithelium and edema, really quite significant. When we gave recombinant IL-4, the urothelium was protected and there was no hemorrhage, but there was still significant edema. If we gave 1 dose of H-IPSE, it looks like a normal bladder. Now, this seemed to be an IL-4 dependent phenomenon because if we added anti-IL-4 antibody with H-IPSE, we started seeing hemorrhage and edema again. Now, histology is always pretty, but it's ultimately subjective. So we also did objective assays, namely measuring hemoglobin levels in the bladder, as shown on the y-axis. So, of course, ifosfamide-exposed bladders are more hemorrhagic. Okay, mesna non-statistically significantly reduced hemorrhage. Recombinant IL-4 reduced hemorrhage, but not in a statistically significant fashion. 1 dose of H-IPSE significantly reduced hemorrhage, and interestingly, there did seem to be an IL-4 nuclear localization dependence aspect to this. And I won't show you the data because of time, but we also saw significant effects in terms of pain from hemorrhagic cystitis being alleviated by IPSE. So there's a very sophisticated assay called the voided spot on paper assay with mice, which is exactly what it sounds like. So mice are prey animals, so they like to pee in corners because they don't want their urine to be detected by predators. Okay, so a saline-treated mouse will void in the corner, okay, just once in a short period of time. But if they're exposed to ifosfamide, they start having such bad bladder pain, they'll start voiding in the middle of the cage and having urinary frequency, essentially. Now, mesna does protect against this voiding dysfunction. Recombinant IL-4 partially alleviates this phenotype. A single dose of H-IPSE also restores normal voiding. And this is IL-4 dependent because if you add in the anti-IL-4 antibody, you start seeing voiding dysfunction again. So this is essentially one of the first examples of therapeutic exploitation of a schistosome protein. And certainly it's the first time that a uropathogen molecule has been exploited therapeutically. We're very interested in seeing whether the anti-inflammatory effects of IPSE can be used in other pediatric relevant diseases, for instance, for MS, inflammatory bowel disease, and even chronic pain syndrome. So I'd like to thank my former and current lab members, especially my senior postdoc, Everest Spinetto, our collaborators, Franco Falcone at Nottingham and Ted Jardetsky at Stanford, our funding sources from the NIH, and thank you for your attention.